just like to tell you a little bit about the Conservation Communities Project, because this project has been funded by the lottery and it's being hosted by the Devon Wildlife Trust. But part of the project is working with Devon Biodiversity Records Centre, and the idea of that is that we work with 11 parishes that have been identified as having very few records submitted to the Records Centre. And one of the reasons they wanted to target these 11 parishes is because they would like to rewrite the biodiversity audits for each of those parishes by the end of the project, which is December 2022. But what they want to do is to use real data from the ground that has been recorded. So part of this project is to get the communities of these parishes recording wildlife, whether it's everyday common species or whether it's something really rare. They would love to hear about it. So if you can spare any time when you see some wildlife, whether it's something from your garden or something on a walk, then please do send in those records so that we can start building up the data. We're holding a whole series of events so that we can help people learn how to identify different species. And hopefully people will then go on to share that information and knowledge that they have with other people in their communities. We're also able to help people improve the biodiversity of their parishes. So if they've got some projects that they would like to have help with to improve the biodiversity, then we can help with that. One of the things I've done as part of the project is to invest in a couple of trail cameras so that people can start recording what's in their gardens to start generating some data from that. So we've got various different ways that we're helping people to gather the data that they've got. Now, I just wanted to tell you something, because over the last couple of presentations that we've done, we've asked people if they've been to one of our presentations before, and most people in the early days hadn't been to one of our presentations, but we're starting to get repeat visitors now, which is fantastic because we're building up a following of our talks and people are learning how to identify all these different species, which is fantastic. But also we've asked the question, have you ever submitted a record to the Devon Biodiversity Records Centre? And the numbers aren't tallying up. We're finding that people are coming along to the talks, but they might not be recording any of their species. So I just wanted to tell you how easy it is to try and encourage you to spend just a few minutes. We had a little test run the other day to see how long it takes. And I asked a volunteer who'd never put a record on to have a go. And it took her just 3 minutes. So if you can spare 3 minutes to make a record of a single species, it will make a massive difference to the data that we hold at the Records Centre. So if you do go to the DBRC website, it's very easy. The third box down in those orange boxes, it says wildlife records. And the first line of that is a link to a form. And you'll see at the bottom of my screen here, there's a form, and that's the number of questions you will have to answer to submit your record. Probably the hardest thing you'll find is trying to locate the exact grid reference. But on their website, they've got a map, so it helps you to identify exactly where that record is. So if you want to just give it a go, don't be scared, be brave, even if it's a house sparrow from your garden or a frog, perhaps, after you've listened to today's talk, we'd still like to hear about it. Every wildlife record counts. If you want to find out more about the Conservation Communities Project, there's lots of information on the Devon Wildlife Trust website. There's a whole project page there that's got information about how to make a record, how you can borrow the trail cameras, how you can get involved in other ways. And also there's a list on there with all links to the various talks that we've already held as part of this project. So the hedgerows, twig identification, tracks and signs, and today's recording will go up on there too. So if you do want to drop back into the website and pick up some of those other bits of information, then feel free. So that's about all I've got to say about the Conservation Communities Project at the moment. I can't wait to learn about the frogs, and Alice is going to take over. I'm going to let Alice carry on. So thank you very much for coming, and thank you, Alice, for coming to talk to us tonight.
Thank you, Jo. I am just sharing my screen, making it nice and full screen. Can you see that one okay?
Yes.
Fantastic. Okay, thank you so much for introducing me. The project sounds awesome. I've used DBRC to upload Newt findings before, so I can definitely vouch lovely and very easy to do. So I'm Alice. Today I'll be talking to you about how to identify Devon's amphibians and my PhD research, which this year I just summed up as the Southwest Frog Swab. So I'm a University of Exeter PhD student. This is me, I want to say maybe 10, 12, 15 years ago with my first pond in my garden, still going strong. I am the secretary for Cornwall Reptile Amphibian Group, the Amphibian Survey Officer for Devon Reptile Amphibian Group, and also a trustee for ARG UK. So I'll make lots of references to these different companies, establishments throughout the talk as well. So unfortunately, you may have heard about this, although our amphibians have survived 4 past mass extinction events, they're not doing so well right now. And the IUCN, which classes how animals are doing, have actually said that there is more than 40% of amphibian species are threatened with extinction. So it's not good. And as they play roles in terrestrial and aquatic environments, because they live in ponds and on land throughout the year, if we lose them, it'll have a big impact on multiple environments. Now, a bit of a warning, there is a spider picture coming up. This is a really nice infographic to show what will happen if we lose our amphibians. As they are predators as well as prey, if we lose them, there'll be a lot more invertebrates, which some may view as really good, but unfortunately this also means that the spread of diseases that spread invertebrates, things such as malaria, could increase as well. So they're really important members of our ecosystems. So in the UK as a whole, we have lots of different reptiles and amphibians. We're just going to focus on our amphibians. So we have our great crested newt, very well known, our smooth newt, our Palmate Newt, our normal toad we're all used to. This is our common toad. This is our little natterjack toad, much smaller. This is our common frog, my current study species. And unfortunately, all of our species that we find here are deemed as threatened largely through habitat loss. And this has come about through multiple different reasons, but largely by sort of human activities, our activities. So for example, agricultural intensification, so the need for farming. We had a big boom around Second World War. That unfortunately means that we have lost a lot of our ponds, although now we are doing lots of work to restore them, which is brilliant. The reason I've left one species off there is because up until recently, we had 6 recognized native species, but our lovely bullfrog was reintroduced in Norfolk. Amphibian Reptile Conservation trusted this, helped rear the tadpoles, which is amazing. So now we have 7 native species recognised. Our local species, however, we've got slightly fewer down here, but doesn't mean they're any less brilliant. So for Devon and Cornwall, we have our common frog, our common toad, and the species you'll see the most sort of well-distributed newt is our Palmate Newt. So those are our local species. Just to go out and identify our species, luckily they are quite distinct, so we can see the difference. This is my favourite amphibian guide. That's the reason I've referred to this one, just because it's got lots of different pictures. You can find loads of other ones online, buy them in shops, and you can print these off or get laminated versions. So this is just the ARG UK's version, made with Amphibian Reptile Conservation Trust. I have this downloaded on my phone so I can refer to it quickly, and it has very nice ways to introduce it. So I'm just going to go through our ID guides on how to see what species we have. So to describe what we found, if you want to upload them as a record, or just if you want to know what you found that day, you can use these specific identification features. So I'll be calling them out, but it helps you to use parts of identification guides. So I don't know if you've ever heard of a dichotomous key, bit of a big word. Basically, it's just using these ID features, as you can see here, this flow chart, to narrow down using these features to see what you've found. It's usually at the front of ID books as well. So for our common frog, I've got this nice big page from that guide, so we could have a little look, and then a picture from one recently in the field as well. So you can see a 2021 frog in all its glory. So our common frogs, as you can see here in the pictures, their colour really varies. I'm used to seeing sort of greeny, browny frogs, but this year I saw reds, so they can really be variable in their colour. Some very nice, easy things to point out with our common frogs. Here, there is a common frog on top of a toad, just so you can see the difference as well. I like to focus on the frog's eyes. Often you can only just see them outside of the water, or if you've got a toad and a frog close to each other, this is a really nice way to do it. So you can see here, this chappy here, this nice big zoom in on his eye. Frog eyes tend to look a bit like sideways rugby balls. So they're not completely circular. They look a little bit like a rugby ball, and you can see that when the head's out of the water. If I go back to this chap here to give an explanation, this is a male. You could tell this as well if they're in the water out on the land. During the breeding seasons, males can actually get quite blue underneath. So if there's a male on top of a female, you can tell them apart that way. And if they're not blue, it can be grey or quite pale as well. If you look at his little front hands here, they have these really dark patches on their thumbs. These are known as the nupital pads, and these are really useful for grabbing hold of the female. So if you do find a frog and you can't tell which is which, those can be really useful to see on the front. And also the males behind their ears have an eardrum, as it were, and that tends to be, let me get this right, similar size to the eye or larger in males, whereas in females it tends to be smaller. So that's another way if you can just see their head. And really good way if you can see their whole body, if they're swimming about. If you have a look here on their back legs, I remember working for Frog Life, they referred to these stripes as their racing stripes. So even if you see frogs in movement, you can see these stripes, and they've also got those nice chunky back legs because frogs leap and jump, whereas toads waddle. So if they're out and about in movement, you see jumping, it's usually a frog, and yeah, they've got these big chunky back legs with those stripes. Okay, our common toad. So I've just gone through our common frog's eyes. You can see here, this is the common frog on top. That is more of that sort of rugby ball shape. Whereas if we go down to the toads, and the same on here on the guide, they look a lot more, I compare them a lot more to goats. So they have, the pupil goes straight across their eye and can be quite thin as well. So it's really squished. And they often have quite golden eyes. So if you see them in the pond, you can look at their eyes and get a good idea of what it is. One different feature as well that people usually use is that frogs are nice and smooth all over, whereas toads are usually a bit more bumpy or warty, it's referred to as well. And yeah, as I said earlier, the toads will sort of squat down and waddle, whereas your frogs will jump a lot more. And toads, if you ever see them outdoors or you startle them, they usually squat right down. I don't know if you can see it clearly in this photo here, but behind their eye, they have a gland which releases a toxin which makes them taste bad to things like cats, dogs, foxes, anything that might lick them. So their defense is that they squat down and release this toxin, whereas you'll usually find if you disturb a frog, they'll jump away. Here's a common toad in the water, just so we can see. I'm going to go back here to show you just quickly. The frog's snout, as it were, is quite pointed, and you've seen this photo here on the left, whereas the toad's a bit more rounded. So if I go to that image of him in the water, nice round nose, you can see that nice little slit pupil. It doesn't have those racing stripes on the back legs either. So these are in the wild photos. And underneath of a toad, if you did find them like this, they don't seem to have those sort of really, really large nupital pads, and they're still bumpy underneath as well. So even if you have them whichever way around, they're bumpy. And I don't believe I have a photo of toad spawn of my own in here yet, but if you have a little look on the identification guide pictures, the toad spawn is actually laid in strings, and this is often found at the bottom of ponds or in vegetation. So you may not see it as regularly as you would do frog spawn. And breeding pairs. So this year, I was out doing fieldwork for my PhD, found some quite interesting pairs of male frogs on top of female toads. It's quite fun for identification to see the difference, but this is just the underside, so you can see what's going on when you see them together in the pond. The males, whether it be male toad or male frog, hold on really, really tightly to the female. And this hugging action is known as them being in amplexus. So this is their sort of breeding position. Unfortunately, a lot of males, both species can get to the ponds first or find a female on the way, and you can sometimes come across things, toad balls we refer them to, big masses of toads. Unfortunately, in some of these situations, if they get into the water, the female may die, but it's not all the time. So it can be quite alarming to see, but if you can leave them be and not try and split them up, that's good. But yes, unfortunately, there can be sometimes too many males at once. But yes, this is one of the interesting pairs I was referring to, is this is a toad and a frog together. Onto our newts. This is our most sort of common species here in Devon that you'll find in garden ponds and right up on Dartmoor as well. I'm sure they're up on Exmoor as well. This is our smallest native newt, the Palmate Newt, and they get the name the Palmate due to the male's nice webbed back feet. So if you can look in this bottom image here, this is a really nice example of a Palmate Newt. He has these fanned out webbings between his back toes, as it were. You can see it in the top image there, but not as clearly. And this is a bit like a palmated leaf, so when all the lobes are attached together. And I mention them because if you see them in a pond, these little black back feet can actually look like they've got little boots on, and it can be really easy to identify if you've got Palmate Newts. And in addition to this, we also have the hair-like filament at the end of their tail. I can see my camera's frozen, but I don't think my screen has. Is my screen all right, Joe? Perfect, I'll keep talking. So they have a hair-like filament right at the base of their tail, you can see here, and that, when they're in the water, is really nice and easy to see if you've got a male Palmate Newt. And when the males fan their tails toward the females, you can see this quite clearly. Just highlighted it there and highlighted. And with both sexes of the Palmate Newt, the male and the female, they tend to have yellow or orange bellies. But the females are a little bit more difficult to tell apart if you're confused between what new species you have. The clearest way to check is that the females underneath their throat, this is males and females, don't have any spots. They can be creamy or pink. Whereas if you had them, you can see there, whereas if you had them next here, a Palmate Newt on the right, the smooth newts actually have little dots all the way up their throat. So that's how you could see if you had a smooth newt. And again, if you want to have a little more information on the other newts that we can find, so the smooth newts, if you have a little look at these identification guides, it goes through a lot more of the features to tell the difference between them. Moving on to our sort of celebrity newt, this is the great crested newt, much more rarer than the Powermate Newt, but still present. And I would show you with my hands that they can be sort of across 2 widths of hands. They get up to 17 centimeters, and they are heavily protected against disturbance by law. So if you wanted to survey these guys or go out looking for them at their protected sites, you would need to be with a licensed person or licensed to do so, but I'll mention that a bit more later. Nice way to identify these guys, if you do happen to come across them, say by accident in your pond, the males have a large crest along their back, which is where the name comes from. But this breaks here right at the tail base, whereas in smooth newts, the crest goes all the way along the back. And they also have a nice white streak in their tails. If you are near a pond and you look in and you see these white flashes, there may be great crested newts in there. Like the toads, they've got warty skin, so they did used to be known as a warty newt as well. And both the males and the females have these lovely golden rings on their toes. So if you can only see those little flashes, if you look out for these rings wrapping around the toes, it looks like they're wearing lots of gold rings. And yes, if you flip these guys over, they have a very bright stomach, and sort of yellow or orange, but with black dots. Okay, we do also have some non-natives present in the southwest. So this chap here, you may notice, is bright blue. This is an Alpine Mute. I put an arrow there to our Palmate Newt, simply because these are non-native species, so they're not classed as sort of being from here originally, but they are a non-native invasive species because they can actually spread disease to our natives and can sort of outcompete them in ponds as well. So if you do ever see one of these, it's good to report them. Locally, Sing's quite a good place to report them to. And I've put the details up on the side there as well. I can see that my Zoom isn't working so well, so I hope that's still recording okay. There's a bit more on non-native species and just with the Sing information there as well. So how to see amphibians. I'm not going to go through a lot of the detailed searches today, just because a lot of them require training. So this is what you guys can do anytime, anywhere, is to go out and see them. So we have daylight visual searches for their eggs. You can also do torch searches. And as I've just said, yeah, other survey methods can require a bit more biosecurity measures and training when things like licenses become involved as well. So for basic biosecurity, if you visit arg.uk.org, it's a really easy one. This will just go through how you can clean your kit. So if you're just doing a visual search or a torch search, you just need to make sure that things like your welly boots are clean, because you won't be using any equipment that will go in the water, but we need to make sure we have clean equipment between all sites. This is for moving things like invasive species, say if they're on your boots, but also most importantly for amphibians, as I mentioned earlier, they're threatened and large by disease, and this can be spread around really easily. So it is best to clean our equipment to make sure we're not spreading anything. How for us to see amphibians in the field nice and easily. Wherever possible, so this is just because we haven't gone through all the training and the different survey techniques. So I think this time of year it's best to look but don't touch. And we can observe a lot of amphibians in the wild. The best thing to do is not to disturb the amphibian eggs themselves. So this is some spawn I took a photo in January down in Cornwall. That was nighttime. This is our daylight searches. So this was a Devon Reptile Amphibian Group identification and survey event just before lockdown last year. So this was March 2020. And you may think, oh, I can't go out in the evening. Well, luckily you can still see a lot of amphibians during the day. So this was in a nice pond up towards Newton Abbot Way. And we were able to see 2 newt species here, and we could see them clearly in the day and in the night. So you aren't missing out if you can't do one or the other. And this is our daylight search. So I hope you can see here. Right in, so if you go to the middle of the screen and then up to the left, you can actually see a Palmate Newt there. So there's a male, we can see a bit of his belly, a bit of his leg, and then a bit of his tail as well. They're very well camouflaged against this, this grass vegetation, but you can see them. So daylight searches are really easy. It's basically walking around the pond, looking amongst the vegetation for any signs of activity. And also you might see things like spawn right on the surface as well. This is torch surveying. Now I always say if you're going to do a torch survey, try and visit the site in the daytime first, or at least get there just before sunset so you can have a good look. So this site, for example, was lots of grass tussocks that kept taking us out on the way to the pond. So luckily we'd been there in the day to sort of navigate our way around. Now, all you'll need for this, I did have some nice equipment to show you, but our webcam's decided not to work. So all you'll need for this is a head torch and a handheld torch. If you have 1 or 2 is fine, absolutely fine. I've got quite a nice big torch now, so it's nice and high powered. And that also has a battery in it, so I can make sure that all my devices are charged up, because you want a mobile phone on you for any surveys. It's always good to have a buddy system in place as well. So say text a friend or colleague or survey group member as well, so when you're going out and when you're back, or at least when you're expected back, for safety. And yeah, so this is us basically scanning the water surface, looking for activity. I was speaking to Joe earlier, I found that frogs are quite skittish if you're out looking for them. Down here, I'm left of Dartmoor, so our frogs have moved on for the year, but you see them oftentimes throughout January into February, and then as we go up country into March, you might be able to still see them and their spawn. And yes, they're skittish. If you sit and wait at the side, oftentimes they'll pop back up if they were disturbed on your first visit. Toads don't seem as fussed. They can be seen on the water surface swimming around in the bottom, and you can also hear them as well. And newts is the same sort of thing. They'll be swimming amongst the vegetation, looking to lay their eggs or breeding activity as well. So this is from a torch survey. This is some frogs spawn that just broke the surface. But I was able to see quite nice and clearly with a nice torch. It was almost sort of daylight levels there. And this is some spawn nice and close up. So I've just said, yeah, if you look but don't touch, and you can actually, this is great for families, rather than taking anything from the wild, it's really easy to observe amphibian growth in the wild. So this was some spawn that I had a nice little look at and thought, well, actually, they've developed quite well. So frog spawn goes from full stop into these little commas and then grows on from there. But you could see that they've just grown out of that nice full stop size. So this is how you can watch their growth in the wild. Something called a Gosner staging chart. So you can use this to actually see the different stages of the frog development. So I've popped down here that the sort of commas I could see in A and B there look-ish like they're from stage 15, maybe through to 17. If you have a nice Gosner staging chart, you can actually go out and see what stage your tadpoles are at. This continues into the 30s and goes, the staging chart goes all the way up to, I think, stage 40, maybe a bit more, when the frogs will be leaving the water. So this is a really nice way of going out in the wild, finding amphibians, this is frogs specifically, and seeing how they grow without having to disturb them, put any nets in the water or anything, because luckily our frogs like to lay right at the surface of the water. Now for newts, their eggs at this time of year, we will be expecting to see them, our palmates as well specifically. So this was taken locally just about 2 weeks ago. Our Palmate Newts and all native newts actually wrap their eggs inside leaves. So they'll use their back legs and sort of fold the leaf in half. It's a little bit like a taco or a nice food wrap, and that keeps them safe. So here you can see this is all wrapped up there. Can you see that there are actually newt eggs? And this is just taken from above the water surface. So again, no need for me to do a dip netting survey or any of the license surveys. I was just able to observe from outside the water and look in and see all these little newt eggs. This is from that nice guide I mentioned at the start. You can see actually how the eggs folded in, differences between newt eggs of different species. And then you might often see these from sort of this time of year onwards, right up into the summer. You can see the newt larvae. And they're just like tiny little newts, but they have these external gills for them to breathe with. I don't know if anyone's ever seen an axolotl, but they basically look like a tiny version of that. So they have these little gills, and they vary in size between the species. But yeah, they're often found in ponds throughout the year, so they're really good if you're looking in to spot them. How to get involved, how to do surveys. So I know you guys are all part of a lovely community project. But if you're looking for any extra training or to come out and actually conduct the surveys or know where they're happening, this is just a nice, easy way to get involved in Devon. So it's Devon Reptile Amphibian Group. That's the website there on the left. And this is the Facebook page, very nice and easy to find. So you can see if there's anything going on, anything you can sign up to, and there's often lots of discussion about finding amphibians and reptiles in your garden or in local nature areas, and people post photos. It's great to just sort of be on the Facebook page and have a look. If you're from wider afield as well, there are these R groups, these amphibian reptile groups right across the UK. So we have Cornwall down here, and then they carry on all the way through. We all tend to have our own websites and Facebook pages. You can access them even without being a member. The reason there I've put members equals ARG web is that we just have a new service where you can actually see all the different maps and things for your areas, the different places to survey, but that's within a website that I believe is only members only. So if anyone's interested in really seeing how your data looks, being able to look online and look at lots of different versions, see it all mapped out, you can become a member, but you obviously can still have a look at everything else without doing so. Luckily now with the rule of 6, it does seem like we will be able to start doing surveys soon. It's dependent for every ARG, but I believe Devon Reptile and Amphibian Group will hopefully be doing some surveys this year, just in small groups. And now right back to the start, what Joe's talking about, documenting and recording our findings. So my colleague Becky Turner from Cornwall Reptile Amphibian Group and from DRAG yesterday did an entire talk specifically on recording. That's for reptiles and amphibians, and that is actually on the Cornwall YouTube page. So if you did want to have a look, that's there. And also for anyone with children, loads of the ARGs now have got YouTube pages where we've put up lots of our seminars. ARG UK themselves, the company, the organization, sorry, have actually done a seminar series which goes through loads of different topics like invasive species and recording. So please have a look on there if you are interested anymore. And also, if you are still teaching from home or like to keep up to date with all these different types of learning amphibian identification, Amphibian Reptile Conservation Trust actually just did some, throughout lockdown, did some bite-sized sort of learning courses, which are all available online. So do go and have a look. Obviously, different preferences for different companies. You guys, it would be great if you can upload your records to iRecord or DBRC for your areas that don't have too many records on there. Luckily, all of these different sites, most do sort of feed in to larger things such as NBN, the National Biodiversity Network. And this is just a page from the Devon Reptile Amphibian Group page. So this isn't, you don't have to use this one. I thought it was just quite a good example. Like Joe was saying, it's only 3 minutes on DBRC, which is fantastic. So yeah, the location you're at, the date and time, really great to take some photos. But I've popped from a safe distance there in case you are, say, reptile surveying, but for amphibians largely, safe distance from the edge of the pond, just in case. So you need to time, the date, location. Photos are great. For the Devon Reptile Amphibian Group, there is the web page there, and also for Record Pool, which is a wider database for reptile and amphibian records, but again, loads of different ways to do that. As well, I mentioned the online pages. You can also see what local research is going on and how to get involved. This was a slide from Becky's talk yesterday. This shows the absolute plethora of different sites you can use, but I've made DBRC nice and big there in the middle. But yes, this was from Becky Turner. If you do want to watch her talk on recording, that is on YouTube now. It's absolutely fantastic. Oh, so I mentioned local research. This is where the talk gets a little bit more sciencey, but it should only be another 10 minutes or so, so it's not too long, but it is my PhD project. So this is actually all local. This is across Cornwall and Devon, and my PhD is called Establishing the Links Between the Skin Microbiome, Pollution and Disease Susceptibility in a Native UK Amphibian Species. And that amphibian species is the common frog, held here in my hands earlier this season. These are all my supervisors, my funding body, and yes, I'm University of Exeter student. So we mentioned earlier that unfortunately amphibians are really threatened. One of the main causes of this is pollution, as well as disease. So this is what my PhD focuses on. So our problems in the UK and for amphibians all around the world, there have been a multitude of different diseases, and we call these emergent infectious diseases, EIDs. So you may have heard of the chytrid fungus. That's a fungus that can grow on the skin of amphibians. Another really big one that's affected amphibians worldwide are rhinoviruses, and we actually have rhinoviruses here in the UK. They've caused amphibian die-offs right around the world. These are some of the places. And all our native amphibians are susceptible to infection, but it largely has caused mortality, so deaths in common frogs, and it's been happening since the 1980s. And this map on the right is from previous research that looked right across the UK, and the Frog Mortality Project, if anyone's interested. That's a great research project. This is one of their maps of the emergence of rhinovirus, where it was found. So in the UK, we have 2 different types of rhinoviruses. We have frog virus 3, FV3-like, and the common midwife toad virus. My PhD is on the FV3 variant, which is the sort of more common one. When an amphibian is infected in this and it becomes a larger, full infection, as it were, we call it rhinovirosis. So if you were to go out and look for signs of this, or if someone was reporting signs of this to you, it can be seen as mass die-offs, so lots of froggies dying at once. Isn't always down to rhinovirosis, obviously that can happen for different reasons, but what makes us think sometimes that it is, you may be able to look out for, is they can have skin reddening or ulceration, or signs of internal bleeding. So I won't put any photos of that, is near dinner time, but if you did think that you have seen signs of this, just with all records, obviously it's great to have records of occurrence when you find them, but if you are seeing signs of disease, any types of disease that look a bit odd, or rhinovirus specifically, you can go to Garden Wildlife Health. They are based at the Zoological Society London Zoo, and they do loads of research across loads of different animals, not just amphibians. So signs of disease, if you can upload them there as a record, that is fantastic. So although we found rhinovirus in loads of places around the UK, it does seem to be occurring in some places without actually causing these mass deaths. And this has made researchers that come before me, so this chap here in the bottom, Lewis Campbell, you can see there, him and my primary supervisor, Dr. Xavier Harrison, have done lots of research on these other factors that could be influencing why we're not seeing these mass deaths. So things that could be infecting why some frogs are really susceptible and others may not be. So this is the main part of my PhD. This is the importance of the microbiome. This is just the community of bacteria, fungus, other things that are actually found on amphibian skin. So we as humans have microbiomes on our skin. You may have seen a lot of advertisements for good gut microbes, for our gut microbiome health. So this is a really flourishing area of research, but amphibians are really special in that their skin microbiome can actually help them in defense against disease. We can as well with our gut microbiome, but this is a sort of a hot area of research in amphibians, that their skin could be helping them in defense against disease. And this is because some of the microbes on the skin can actually release these chemicals known as peptides that can fight off the disease. Can also be through competition. So if you've got loads of different species on your skin, there may not be space for the disease, fungus or virus to even get in there and establish because of this competition, not just through space, but also through this release of chemicals. So mentioned again, previous study, this was Lewis and Xavier's work, have found that the more diverse, so the more species of microbes, say bacteria, on common frog skin can make them more resistant to rhinovirus. So if they've got loads of different species on their skin, this seems to help them in not getting rhinovirosis, so that sort of severe infection. And yes, just as in humans, we have probiotic yogurts. We can do transfers of gut microbiota. There's even sort of fecal transfers. For amphibians, there has been some research in moving microbiome bacteria from the skin of some amphibians over to other species in order to help protect them against disease. But it's largely focused on that chytrid disease I mentioned earlier. I won't go into all the different types of chytrid, but yes, most of the research has been focused on that so far. And my PhD is looking at combining the microbiome, this disease rhinovirus, and also the influence of pollutants. So I've made this nice little diagram here to show this sort of connected pathway. So amphibians, I've popped permeable here, but basically that just means their skin isn't sort of skin tight, stuff can get through it, pass through it like water and air. This means that they are really vulnerable to pollution. So if they're sat in a pond, those pollutants can go sort of straight into their skin and into their bodies. And pollutants, those that are antimicrobial, it's within the name, can kill, take out microbial species or just lower the diversity. And as we've just learned, they need those different species either for competition, or there may be some in there that are key species that are producing those nice chemicals, those peptides. So if a pollutant is lowering their numbers or killing microbes, then it can actually alter the amphibian's response to disease, so their pathogen susceptibility. Now, Lewis did some amazing research on this, and he found that at different populations of frogs throughout the UK, there was a different microbiome diversity between populations that had Rhinovirosis, so the actual outbreak of rhinovirus, compared to sites with no outbreaks. So in some places, rhinovirus was present, but it wasn't causing rhinoviruses and those big die-offs. And they think that that could be linked to this diversity of microbes on their skin. So yeah, to sum that up, observe microbiome variation, so that difference across populations could be linking to differences in rhinovirus infection survival. So this is huge. This could be a way of finding out what's going on with this disease. So research is required into our UK common frog populations to understand the links between the microbiome, pollutants, and what's going on, the diversity, the structure, the variation of their skin microbiome. And yeah, this could explain the differences we're seeing in rhinovirus infection. So just very quickly, I'll be going through what I will be doing. I'm looking at the links between the skin, the pollutants, and the microbiome, just to see if this sort of gives us an idea of if this is one of the reasons that we're seeing those differences in infection outcome, and if it could be used for future conservation efforts as well. So I'm looking at this is my overall aim. I won't read that all out, it's just a summary of what I've been saying, seeing if it's modifying the skin microbiome structure. This is a nice infographic of swabbing a frog there to give you an idea, to have a swab to show you, but my webcam is down, unfortunately. So post COVID-19, I had to stop field work and everything last year, but this year I've managed to get out in the field. I'm doing a literature review, looking at the microbiome and wildlife health, and I've been doing field surveys. So I've been doing this since my first trip out was in November down in Cornwall, but largely in January and February. I swab the frog skin, as you can see there, and that is to look at the difference of the microbiome diversity, so it's variation across the spatial scale. So I've gone for Cornwall and Devon, multiple sites across there, to see what species of microbes are popping up and where. I'm swabbing the water because this allows me to see the environmental microbiome, so that's what microbe species are sitting in the pond. And these biometric measurements are just simply size and weight and photographing them so that I can refer back to that afterwards. I've also been taking the water pH and the temperature, because loads of these what are called abiotic variables, these environmental variables, can actually influence the microbes that are present. So I'm making sure I've got lots of different variables so that I can look at it as a big whole picture. And then yeah, in further years, so after this year, I'll be doing lab tests. So I'll be looking at combining these swabs, isolate of the disease and then the pollutants, and seeing how they all interact. So I won't actually be sort of testing that on a live frog, as it were. It'll all be in the lab. Right, so this year, I'll go through what I've done so far. On the left there, you can see the equipment I have. This long bit in here that looks a bit like it's got a clock head. Those are my measuring things for actually taking their calipers. I can measure the snout to vent, so the nose to sort of the top of the legs of a frog. These are just some very nice kitchen scales. And as you can see, they've popped a Tupperware pot on top to measure the frogs so they don't hop away. They're very good at that. And gently capture the frogs, swab their skin, like I said before, measured, that's it, the snout to vent length, and weighed them. So this is me just weighing one here. And as you can see, I'm wearing gloves. I didn't go through all the survey equipment because Becky did a fantastic job yesterday, so you can definitely have a look at that. And because we're only doing, I've mentioned visual and torch surveys, you don't need much equipment. However, if you are to find a frog out and about and you're a bit worried about it, or you had to move it, say, out of a road or out of the way of a cat to somewhere nice and covered, I do suggest that you wear gloves. And that's just because our hands have some natural salts and chemicals in them, and that can actually be not so good for the frog's skin. But also, in light of sort of biosecurity and not moving disease around, gloves are best. If you don't have gloves, however, you find a frog and you think, oh my goodness, I need to move it. One really quick and easy way of making sure that our stuff on our hands doesn't hurt them is just to dip your hands in some water, and that creates a nice surface, make it a bit more comfortable for them. So here, this is a nice photo I showed you guys earlier of a toad and a frog. So far, I've come across some interesting observations. So I keep finding frogs on top of toads. If anyone's interested why, I can chat about that in a minute. I found multi-species ponds, which is fantastic because each of these species, so the newts, the toads and the frogs, tend to like different areas of ponds or different types of ponds. Again, I can talk to you about that in a moment if anyone's interested to know the differences. And so you don't always find them all together, but I keep finding in my surveys that these sort of nice big ponds or even small ponds have got lots of different areas of habitats. So shallow area, deeper areas, planted areas can actually host multi-species. So one I went to had frogs, toads, newts, and fish. And fish can eat spawn and smaller bits. So a lot of the amphibians don't breed in the same ponds as we would use for ornamental, but this has been really encouraging to see these multi-species ponds. And also anybody worried about those big freezes we have, I was a bit worried because last year we had that big storm and the cold and it sort of put an end a bit to the season a bit early. This year, however, I've seen frogs active before and after the freeze, and the newts were out nice and early, as I've seen them in Cornwall and Devon, so that's great. This is just me holding the frogs and how I take photos of them. You'll notice that I hold them below their tummies just because frogs don't have ribs. So you've got to be really careful not to squish them. So I hold them sort of around their waist like that. That's them from the underside. And if you see that code on my hand, that was just me. For each individual, I changed my gloves. That's because I'm doing a microbiome survey, not just because of biosecurity. So I want to make sure that each frog that I swab is sort of its own community, own singular sample. So I pop a little number down on my hand to remember each one. That also goes on the swab tube. And if anybody is interested in a project, and I'll waffle on about it, there was a paper came out last year between me, Xavier, my supervisor, and Lewis, so the chaps that did all the previous research, and this goes into a lot more about amphibian rhinoviruses in Europe and current research, past research, and directions for future research. So do have a read of that if you're interested. It's got a nice little infographic there to show all the differences. So for you guys listening, this was my map of ponds last year. Obviously not every pond is active every year, so I've visited some, they weren't active, so I'm also always on the lookout for active frog ponds. This doesn't have to be now, obviously the frogs may have already moved on from your pond, but I'd be really keen to hear if you have frogs, if you'd be happy for me to visit and swab them. And also it's great, like we're saying, recording data. There are loads of places you can pop your data online. But yeah, if you're interested in me visiting, let me know if you're in Devon and Cornwall. And as well, I'm quite interested to see if you've seen any rhanid herpes virus. So again, this is a bit of a lighter image. You can see that they've got this sort of bumps on their skin. I haven't gone nuts and shown you sort of a really, really bumpy one. But if you see bumpy frogs that you think, oh, that doesn't look normal, and it's a bit more cloudy in these bumps, you can go to Garden Wildlife Health, report it there. That should be your first port of call. But I'm also quite interested to see. So if you did want to tell me, that's also absolutely fab. So thank you everyone for listening. Really enjoy speaking about amphibian ID. I am here to answer your questions. You may not be able to see me, but I'm sat here and ready to go. And thank you very much, Joe. Thank you for helping me do this talk in the first place, with getting in contact. Thank you very much, everyone.
Thanks, Alice. It seems strange that we can't see you, but at least you've been able to reconnect and carry on. Oh, you're there. We can see you.
I tried it. It's working. Fantastic.
Brilliant. So we'll just have a look at some of the questions. There's a few here. So the first one says, I live outside the parish that you require. Can I still record on DBRC? So yes, anyone who lives in Devon, or even if you live outside of Devon, but you come to Devon and you've seen some wildlife, then please record on DBRC's website. You don't have to be just from the 11 parishes. That's just where my project is focused. So please do record from all over Devon. That would be great. It says here, this is a question for you. If you see a crested newt in a developed area away from a pond or water, should you try and relocate it or report it, or just leave it?
Okay, so great crested newts. So the licensing with them is largely that you shouldn't disturb them. So that includes torch surveys. And even sort of going up to the ponds, unless you are licensed. If they are in an area, so you can actually check online the Great Crested Newt Buffer Zone. So those are the sort of the known ponds, known areas where they exist. So if you say you found it away from that area, you can ring believe you can ring Natural England, but you can also ring an ARG group and report to them that you found a great crested newt in a new place. But generally speaking, you shouldn't move them just because only licensed people should pick them up. If you found them in a road, and worst case scenario, you can move them to the side, but also all our newt species live on land and in water. So if they're out and about and they're not directly near to a pond, it may just be that they're in their terrestrial phase, they're hibernating, or they're on the lookout for food. So unless they're in sort of immediate danger, say if they're on a main road and you can pop them to the side, the best thing to do is to leave them be. And yeah, if they're in an area where they're not in one of those buffer zones, you can check locally online, see where they are. You can give your local ARG a call and say, I found a great crested newt, maybe a new population, because that's brilliant. That means we can note that down and create a nice buffer zone around it as well.
Thanks, Alice. So Jenny says, would a red torch be better for, when you talk about doing your torch surveys, would red ones be better?
Yeah, so I was going to say, I can actually show you now. I have a handheld torch, which is just normal white light. And I usually wear a head torch, which is red light as well. Red light is really nice for all animals. I believe it's a bit sort of softer on their eyes. If you are happy once you get to the pond to turn red light on, that's brilliant. You can see the eye shine. I tend to do a mixture of both. So if I need to see and identify them, sometimes I find that's clearer with sort of a white beam. But you can use red lights. I often find that if I'm walking to a site, sometimes I can't see as well with red lights. So it's quite nice to have a torch that can do both.
That's good to know, thank you. John says that he's lucky enough to have 2 ponds, and he finds that often the spawn is on the edges and then the pond levels drop. Should he be moving those spawns into the water as the levels drop, sort of to save them, really?
So this is really interesting. Frogs tend to prefer these shallow areas where a lot of the time it does dry up. And that's simply because they like laying their spawn where it's going to get warm. It helps the spawn develop. Also in sunny areas, you get things like algae, and the spawn, the tadpoles will eat the algae. So it is their preferred place, these sort of shallow areas of ponds. If you have sort of a beach area, as the water recedes, it should sort of move the spawn with it. But the spawn does like to be at the surface. So if you've got a deep pond and you moved the spawn further in and it dropped, that's not always the best. So if you can leave it and maybe just top up your pond so that it sits in a little bit of water and doesn't entirely dry out, that's often better than moving it into a deeper bit of the pond. But yes, if you do have a nice beached area, as the water drops, they should go down with it. But if you needed to nudge it, an inch or 2, fab. But they do like laying in those shallow areas, although yes, it can be a bit annoying if it dries up and you think, oh, why froggies?
Quite a lot of people ask the question, don't they, if they see a puddle and there's frog spawn, should they move it, or a ditch that's dried up? What's your thoughts on that?
I personally, I know it's really difficult. I do try not to move spawn, simply because if the frogs laid it there, there's usually a reason that they've chosen that site, and that may be because it's shallow. They believe it's going to warm up nicely and it will develop there. Joe, you and I had a chat before as well, and it is unfortunate when the spawn dries up, but it does provide food for other species. And I think the only time I would ever advocate for moving spawn is, say, if you had a puddle by your front door or to the side of your house and you saw it was going to really dry up, in which case, if you've got a pond, you can move your spawn in there. Some people also put them in sinks, your little plastic sinks. You can pop them in there. But if you ever sort of move them or decide, okay, I've got nowhere safe, I'll pop them in this little plastic tub, you've got to be really careful, because as the frogs develop, towards that late stage, if you look at that Gosner staging chart, their back legs can pop out really quickly, and then they change from aquatic to wanting to be on land, and they can drown. So you've got to keep a really good eye on the spawn if you did have to move it into somewhere that's not like a natural pond with a beach. But yeah, I know it's really tough. I advocate for not moving it unless, that is end of the world. And if you do have to move it, it should only ever be local. And that is because, and I mean really local, so it's not good to sort of share spawn with your neighbours and things. The best way to look at it is build it and they will come. If they're not laying in your pond, try not to move them in. But yes, if you're really, really worried and it's very, very local, and yeah, you can move it, but I must stress very locally, because it can move invasive species and disease as well. So it's best to leave it if you can.
Thank you. That's really good advice. That's one of the most popular questions I think I get asked about frogs and frog spawn. There's a question here again from John who says, how have you selected your ponds for your surveys, to take part in your PhD?
Yes, so previous research I'd done in my undergraduate was all on Dartmoor. So I was looking at newt ponds, but I'd often seen frog spawn. So I'd kept them tallied and recontacted the landowners. I also put out a lot of notices. So through the uni, on Facebook, through pages like Drag, Crag, and then also on like garden wildlifing forums, because I'm sure you guys have all noticed the same. During lockdown, people have taken to the internet to share their amazing ponds and their gardens. So that's been a really good source for me. But largely it is through things like map surveys, looking at where there is water, checking online databases, which is why it's so important that all our records go online, because I can actually see where it is. And then usually I'll contact potential pond owners, obviously with making sure that it's all compliant with GDPR. So if it's Facebook, I can reach out, but then I usually send a form to make sure people are happy with their contact details being saved. And down in Cornwall for my local ponds, I actually did some letter delivering this year with a little thing of, do you have a pond? Have you had any frogs? It was very COVID safe. They all went into little laminated pouches and were posted through the door with me with a mask on and gloves when I prepared them as well. So it's been a bit of a mixed bag, online, letters, and through previous knowledge, but I'm always open for new ponds as well.
Oh, that's great to know. Thank you. Is it only toads that croak? That's from Alison.
Oh, I wonder if I have the app. So yeah, so toads croak. I often think they sound like birds. There is I'm trying to think, the Frog Life app. If you have a little look on that, on the App Store, whatever device you have, there is actually loads of different amphibian recordings. So you can go through and hear all the different noises. So the toads croak, frogs also make a noise. Newts can actually make a noise if you pick them up and they're not happy, or if they're startled, they can squeak at you. Very not well heard and shouldn't be, like if you're holding them properly or ever had to move them properly. But yes, all of our native and our invasive species, frogs and toads a lot more, do make noises, and some of the invasives are amazing. So midwife toads sound like clangers, and it's a really metallic sound. So do have a little look online, and yeah, that Frog Life app is fantastic for hearing all the different noises.
I never knew that newts made the noise. That's great. There's a nice question here. Any advice for young conservationists who would like to do similar research one day?
Yeah, I sort of went on a bit of an odd path. I did art before I did science. I've always been interested in ponds. Basically, I volunteered a lot. I went to lots of different places. I didn't know that I liked amphibians straight away. My granddad had koi ponds, so I was doing surveys every year and we took all the detritus out, and that's how I got into amphibians. I did a diploma rather than A-levels and then volunteered at lots of different places. So Dartmoor Zoo was one of them. And then I did a BSc, and I've done a master's and a PhD, but you don't have to follow, you know, that entire academic route. Nowadays, it's so easy to make things like vlogs, TikToks on what you're doing, go to these different events. I mean, virtually, there is so much out there you can get involved with as well, which is fantastic. And yeah, just find what you're passionate about, get out there wherever possible, either by yourself, with parents, with these groups that I've mentioned this evening, and just try and yeah, find the things that you're interested in. And I find that passion for me has sort of driven me through what I like doing. Done lots of sort of museum volunteering, and so many places are happy to take on volunteers. So I just think, yeah, follow your passion. It doesn't have to be university or courses, but there's so many different ways of doing it in terms of volunteering, just being out in nature.
Yeah, absolutely. We've got a comment from Jenny that she's got a multi-species pond. The frog spawn have survived freezing twice. The tadpoles are out wriggling around already. That's just outside Plymouth, so I guess
fantastic.
Yeah.
Yeah, no, but luckily, I mean, I was a bit worried this year. We did have that big freeze, but the way that the frogs lay their spawn, you know, safety in numbers, as it was, a bit like when penguins huddle. Even if there is a frost, there's always a hope that some in the middle will survive. And you know, they lay loads, and only a few survive every year. So that's fantastic to hear that down here in Plymouth as well, that they've survived 2 freezes. That's brilliant.
And then one last question from Alison is the chlorine in tap water a problem to frogs?
Yeah, so the best way to fill up, if you said, yeah, if you had a frog pond that was drying or a newt pond that was drying, I use the water from my water butt. So that's all naturally from the rain. But if you don't have a water butt and you wanted to use tap water, you can basically fill up a bucket or a big bottle and leave it out sort of for a day or a little bit longer. That sort of reduces that chlorine risk, and then you can use that in your pond. So it's best not to go straight from tap to a pond. If you can, yeah, fill something else up and then use it, say a day or a couple of days later.